Welcome to VodCast 4.2 in our unit study of genetics. We'll begin this VodCast with a quick review of history as to some of the groundwork that was laid for being able to make predictions for future generations of offspring. A lot of this work was produced by an individual named Gregor Mendel. We sometimes refer to him as the father of genetics. He was a monk, and in monk, back at the time in monk culture, oftentimes they lived in a small community, and everyone had certain jobs or characteristics. Gregor Mendel was the gardener, and he did some very interesting things with plants and study of plants. He grew many different vegetables, one of those being peas. Pea plants. And what he did was he grew a large number of plants, but he was very careful to observe those plants over a period of time. And seeing a number of generations over time, he started to begin to make some observations, but he recorded those observations and what he referred to as a trait. And using a very systematic process of math, to analyze those results and sort of verify his observations. And what he arrived at was this foundation of understanding of being able to predict what traits are going to show up in your offspring. Now, for Gregor Mendel's pea plants, there's a number of traits that you could look at for any plants, as far as the height of the plant and the shape of the leaves and the branching of those leaves and flowers and color of the flowers. Mendel happened to choose seven characteristics that he studied of these pea plants. For example, the color of the pods, or the shape of the pods, or the color of the flowers. And after a number of times making these observations and recording these in a journal, he was able to identify these traits and isolated them, literally segregating some of these plants that had these pure characteristics that every time you would reproduce these plants, they would show the same traits over and over again, the same color of the pod, same shape of the pod. And then he would take two of those parent plants with two contrasting traits and produce what's called a hybrid. He would mix them and then see what would be produced as far as that similar trait in the offspring. And he made some really interesting findings as far as what the percentage of the time traits would appear in the offspring. We're going to take a look at an example here in just a moment of one of these traits and how we can use a mathematical process called a matrix, or sometimes referred to as a Punnett square, to help make those predictions. So let's have a look. As we mentioned previously, and for the parent generation, Mendel was able to segregate some of these pea plants and keep them isolated, reproductively isolated, from reproducing with other plants, so that all of the traits that would show up in these parent plants were what we call purebred, or, as we learned in the previous podcast, homozygous, this very similar. Now, Mendel did not have quite an understanding that we do today of chromosomes and chromosome patterns of inheritance and the idea of a gene. He didn't have that background. What he did know was if he had these certain characteristics raised in parent plants and made certain crosses, he could accurately predict what traits would show up in the offspring. He referred to these as factors. So let's take a look at an example here. He would take one parent individual here, we'll call large S as their genotype, producing smooth seeds, and the other parent genotype, small s, or homozygous recessive, producing wrinkled seeds. So the hybrids that he produced were referring to this P shape. In the hybrids, all of the offspring would be produced with this smooth trait. Then he did one other interesting step. He took all of the offspring, we refer to this as the F1 generation. F1 stands for familial, the first generation of those parents. And he crossed those as well. And we can see here, using our little key, that the large S is a dominant trait, indicating smooth seed patterns, and the lowercase s produces a wrinkled seed. We take the two smooth seeds from the F1 generation that were crossed, And we understand that these seeds, as we learned from our marshmallow creature lab, has the ability to produce gametes that contain either the dominant allele, the smooth, here, or the recessive allele, the wrinkled, here. So the key to understanding this process of inheritance is going to show up in this F2 generation, the second time we cross these hybrids, if you will. Because the F1 generation parents have the ability to produce either a dominant or a recessive allele in each of those seeds, we can use this device down here, referred to as a matrix or a Punnett square. And basically, we would take the gametes that are potentially formed from each of those F1 generation individuals, and we put them on our Punnett square on the top and on the sides. So on the top of this Punnett square, we see the two gametes with the alleles, the capital S dominant here, and the lowercase s for the recessive condition here. We also see on this side the other gametes that are formed, again, with a large S allele indicating smooth and wrinkled. We would consider these combinations to be heterozygous. The F1 generation produces these gametes that have a heterozygous combination, a mixture, if you will, of both of those alleles. Now the interesting part is when you fill in your Punnett square. In order to fill in this matrix, we simply take the alleles on the top and on the side and combine them, forming an individual that has the homozygous dominant allele combination, large S, producing a smooth pod seed. We take the other alleles on the top and on the side, producing this individual that is a heterozygous allele combination. But again, possessing at least one dominant trait gives you a smooth seed. On the bottom, same deal. We take the alleles present in the F2 and bring them down, and we see a heterozygous combination here for that seed. And lastly, in this fourth box, we notice that the alleles that are brought forward are both recessive, and we see that this gives that individual the trait of being wrinkled, a wrinkled seed. If we were to count up the total number of results we see out of the 4 possible offspring, 3 have these smooth seeds, and only 1 has the wrinkled. With Mendel's observations in mind about pea plants, we can apply that to most individuals and looking at inheritance patterns that are produced through sexual reproduction. So we use this tool called a Punnett square. And we'll run an example here in a moment to show you how that process can work for a number of different examples. One of the concepts that we want to help get across is this idea of a Punnett square can help predict the expected results of a cross. We're going to make some predictions, and we can be fairly accurate on the results that we predicted. Punnett squares also show which genes can combine when the sperm and ovum meet. If you remember from the last example with the seed shape, we noticed we had certain combinations of the large S, or referred to as homozygous dominant. The large S, small s, produces also smooth seeds. We call that heterozygous. And lastly, the homozygous recessive, which would be the small s. Both of those alleles are recessive in nature. Let's produce a practice problem and example to see how this plays out. If I were to take an individual that was homozygous dominant for seed shape, homozygous dominant, 2 of the same alleles, and in this case we're using a large S to indicate that allele. And I took another parent, which was heterozygous for seed shape, which would contain 1 large S for the dominant allele and 1 small s for the recessive allele. In the previous example, I fill in my matrix by combining the possible combinations from each gamete, and I will fill in my Punnett square here. Once we have our F2 generation filled in for alleles, we could actually take a look and report out the results from our observations. We see here that if I count these individuals up, I have 2 of the F2 offspring that are referred to as homozygous dominant. So I record them here, 2, large S, large S. And then I use what's called a semicolon to, or excuse me, a colon to compare these results to any other results that have showed up in the Punnett square. For the other two offspring, I see we have a heterozygous combination. One allele is dominant, one allele is recessive. And I would record that over here next to my Punnett square. And I see that I have a 2-to-2 ratio, or if I were to simplify, a 1-to-1 ratio. Now we want to take that one step further and determine what would those genes produce for a phenotype, the physical appearance. Here we have a genotype, a 1 large S or homozygous dominant combination compared with a 1 heterozygous combination. That homozygous dominant combination would produce smooth seed pods. We also look at the heterozygous individual, and again, all of those offspring, even though they contain the recessive allele, they still have at least one dominant that are paired with it. So this in turn produces all smooth seeds. We will spend some time later in this activity to allow you to practice various combinations, making predictions for that F2 generation, and creating a genotype and phenotype ratio like you've seen here on the slide. Knowing that genes are found on chromosomes, and also knowing that we inherit 2 copies of every gene because we contain 1 chromosome from each parent during sexual reproduction, there's a lot of information and predictions we can make, as we saw with our Punnett squares. In addition to that, if I were to take a step back and look at the arrangement of chromosomes from one cell of an individual, I could produce this next item, which is called a karyotype. By definition, it's an image of the chromosomes found in a single cell arranged by size. Much like you did with the marshmallow creatures, we used paper strips to represent the chromosomes, and one of the first set of directions was to remove those paper strips from the envelope. We had red and green to represent combinations from both parents, and lay them out on the table, face down, in pairs. We have pairs of chromosomes because, again, we have 2 parents. If I were to take a picture of all those chromosomes before I would arrange them, they would be rather jumbled on the desk. One of the key points of doing a karyotype is to take a cell that is currently in mitosis, which is in one of the dividing stages, and take a snapshot or scanned by a computer and help to organize those chromosomes in the pairings and relationships to size. And we get this picture that you'll see in a moment that will allow us to interpret and make predictions about what is contained in those chromosomes. A few things that we can tell from a karyotype would be something like this. The sex of the offspring can be determined genetically. We could locate the number of chromosomes that are found in an organism. Now, if you remember, for humans, that number is 46. 46 total chromosomes are 23 pairs. That number does differ for different organisms, and it's characteristics of your species. The last thing we could tell from a karyotype is that of those 46 chromosomes, are there any extra chromosomes or deletions of chromosomes or parts of chromosomes, which might indicate a situation like a disease or some kind of genetic problem. So let's have a look.